Hello dear students, this is Mushtaq from MC Society Institute of Pharmacy, Azam campus. Today we are going to learn about mechanism of drug actions. Outcome of today's lecture: the students will be able to state the various types of mechanism of drug actions. Students will be able to describe the various types of mechanism of drug action. So mechanism is how the drug will act in our body. That is called as mechanism of action. So we do not know the precise exactly how the drug will act in our body. So for our convenience, we recognize 5 patterns of drug actions. The drug may act in 5 waves or patterns. First is called as drug receptor interaction, that is called specific interactions. Second is drug acting on enzymes, we call it as partial specific interactions. Third one is non-receptor mechanisms, that is we also call it as non-specific interactions. Fourth one is drugs acting through antibodies, and fifth one is placebo effects. So these are the 5 patterns by which drug may function in our body or drug may act in our body. First we'll start with the drug receptor interactions, that is called specific interactions. So in this image you can see the cell wall. This is your cell wall. In the cell wall we can see the proteins, molecules which has a particular shape, etc. So these large macromolecules of proteins, normally we call it as receptors. So you can see here the receptor. So what we can do is,
most drugs act on a cellular level by chemically binding to receptors on cell membranes or inside cells. The cell membrane contains receptors for many kinds of substances such as hormones and neurotransmitters. Some receptors also interact with drug molecules. As a drug molecule binds to a receptor, reactions are initiated that stimulate.
so we can see here that the receptors are normally located on the cell wall. It may be inside the cell also, but some drug molecules are normal substances that are called as ligands present in our body will attach to these receptors. So you can see here in the video. So once again we can see, so this is a receptor and the drug or a macromolecule will attach, that is called as a ligand, will attach to this receptor to show its functions. You can see here.
As a drug molecule binds to a receptor, reactions are initiated that stimulate or inhibit regular cellular functions. One type of reaction activates.
So you can say here the binding of the receptor with the ligand or drug, and after binding it will initiate an action.
Deactivates or otherwise alters intracellular enzymes. Almost all cellular functions are catalyzed by enzymes. Therefore, drug-induced alterations can greatly increase or decrease the rate of cellular metabolism. Another type of reaction involves changes in the permeability of cell membranes. A drug molecule binds to an ion channel receptor, a receptor protein, that is a structural component of the cell membrane.
So you can see here the drug or ligand is coming and will bind it and show an response.
The drug's binding may open or close the ion channels to allow or prohibit the movement of ions into the cell. In nerve cells, for example, movement of sodium ions into the cell usually excites the cell. Movement of potassium ions out of the nerve cell inhibits nerve cell excitability and function.
So we have seen the receptor and the ligand which will attach to the receptor and shows an action. Just we have seen that it will open the channel. So now by its basic viewing we can say what are receptors. Receptors are macromolecules with which a drug will interact to produce their characteristic biological effects, or we can say a pharmacological receptor is a macromolecule with specific sites to which specific substance like drugs bind or attach. So normally biologically the receptor is active, as we have seen until a ligand binds to it, and then only it gets activated and triggers a post receptor response, that is like opening of the channels. So next is location of drug receptors. The sites in which drug receptors are found are normally on or within the cell membrane. Second, inside the cell, they are present inside the cell. So we can see here in the image, so this is present within the cell membrane. Next, the pharmacology receptors are named according to either first the principal endogenous agonist that activate them, like adrenoreceptors. The receptors are named because the adenine is the endogenous substance which attaches to the receptor, so that's we call this adrenoreceptors. Next, we cholinereceptors. Why we have called it as cholinereceptors? Because acetylcholine will activate this receptor, that's why we called it as cholinereceptors. That means the name is depends on the normal endogenous substance or agonist which is present in our body which activates them. Second is naming will be depend on the first exogenous agonist found to activate them. So for example, opioid receptors, these receptors was first found out by the opium. This opium enters in our body and attaches to this particular receptor, so then we have called this particular receptor as opioid receptors. Same example as benzodiazepine receptors, whether or not an endogenous agonist is discovered later or not. So two naming systems, one is because of principal endogenous agonist, second one is the first exogenous agonist. What will happen after the drug will bind to the receptor? Three things may happen. First, receptor ion channel coupling, that means after the drug will attach to the receptor, there will be opening of the ion channels from which the ions like sodium, potassium will flow on or transfer through across the membrane. Second is receptor enzyme coupling, in which when the drug binds to the receptor, there will be activation of an enzyme, and this enzyme shows particular catalytic actions. Third one is intracellular steroid receptor. Because of this, when the drug binds to the receptor, there will be protein synthesis coupling, that means there will be synthesis of specific proteins with specific codings that will change the activity of cells. So receptor response, there are three types of response. Once the drug binds to the receptor, it may activate, some drug may activate ion channel opening, some drug activate enzyme, some drug activate protein synthesis. So we will see a animation.
There are 4 types of receptors: ligand-gated receptor, enzyme-linked receptor, G protein-coupled receptor, intracellular receptors. Receptors have specific site to bind to molecules or ligand. This is called a lock and key model. In ligand-gated receptors, the cell membrane requires specific ligand-gated channels for movement of hydrophilic molecules through it. First, ligand binds to the receptor, then the receptor gate opens. For example, acetylcholine binding to nicotinic receptors. In this example, sodium enters the cell and electrochemical gradient is created, resulting in depolarization. In enzyme-linked receptor, the transmembrane protein of the receptor are enzymes, or closely associated with enzyme. For example, tyrosine kinase receptor. The binding of the ligand induces dimerization of the receptor, which activates its tyrosine kinase activity through autophosphorylation and phosphorylates other intracellular enzymes sequentially. The phosphorylation status of the enzyme may result in activation or inactivation, resulting in various cellular responses. With the gene-protein coupled receptor, first the ligand binds to the receptor, which causes structural changes in the receptor and in turn changes the shape of the G protein, which reduces GTP to GDP. This leads to the separation of the alpha subunit from beta and gamma units. This in turn activates other enzymes and effector proteins. Intracellular receptor. Ligands that pass through the cell membrane enter the cell and into the nucleus, crossing the nuclear membrane. After entering the nucleus, the ligand binds to the intracellular receptor. This can alter the transcription of DNA within a cell.
So normally we have seen the receptor types and its responses. So ligand gated, G protein, kinase linked and nuclear receptors. This receptor mechanism is the only clear mechanism. In this we have to understand two terms. First is the ability of the drug molecule to bind to that receptor is called as a affinity. If the drug has affinity, then only it will bind. If the drug doesn't have affinity, the drug will not bind to the receptor. Once it is attached to the receptor, next is its response, its action. So the capacity of the drug to give rise to pharmacological action after binding to the receptor is called as efficacy or intrinsic activity of the drug. The drug which has more eccentric activity, that means high efficacy, the action will be more, the receptor will be stimulated more. So two terms, affinity and efficacy. The ability to attach to the receptor is called affinity, and then create the response is called as efficacy or intrinsic activity. So depending on this, we can create on a one receptor we can create 5 types of drugs. We can synthesize 5 types of drugs, and these 5 types of drugs we can categorize into: first is agonist, which shows the same action; antagonist, which shows the opposite or stops the action; the partial agonist, which shows the off action of the agonist; mixed agonist and antagonist. In some cases it shows agonist action, in some cases it shows antagonist action; inverse agonist, which shows inverse effect, means opposite effect. So drug attached to receptor site and may produce or prevent a response. So we can categorize into 5 types. Individually we will see one by one. First is, we show a animation agonist, which shows a full action. When the drug binds to the receptor, it shows a full action as normal endogenous substance, that is called agonist. Next, drugs which show actions are called as partial agonist. In this partial agonist, drugs may show partial action, not as a full action. That is called half action or less than full action. This is partial agonist. Third type is antagonist, which blocks the receptors. There is no effect. So it blocks the receptor, so the agonist will not attach to this antagonist agent. So it's totally blocks the receptors. So we have seen the animation of agonist, partial agonist and antagonist. So we'll see one by one. An agonist is the drug which combines with its receptors, activate it and initiate a response. Examples are acetylcholine and non-adrenaline. Just we have shown the same action will be produced by a drug as which is seen by the endogenous substance like acetylcholine and non-adrenaline. Next is partial agonist. As we have seen in the animation, it will not show a full effect, it show a half effect or less effect. So a partial agonist will bind to the receptor but activates it weakly and prevents the action of a full agonist. So in fact, the drug shows as action on the receptor with an intrinsic activity or efficacy less than 1. Antagonist is a drug which binds to the receptor but do not activate it. However, it prevents the action of agonist by rendering the receptor unavailable. Examples are atropine antagonizes acetylcholine, phentolamine antagonizes non-adrenaline. Another term is the pure antagonist. A pure antagonist has no action of its own, just it will block the receptor. Examples are opioid receptors agonist, that is naloxone. Third, inverse agonist, which shows an opposite action as same as agonist. So the action will be inverse that is opposite action, that is all called inverse agonist. Examples are GABA receptors antagonist like benzodiazepines, etc. Some drugs are there which shows an opposite action which causes epilepsy, etc. because of stimulation of the drug. And fifth type is mixed agonist and antagonist. A mixed agonist and antagonist is a relatively new term and it's frequently confused with the partial agonist. So a mixed agonist and antagonist is a drug that activates simultaneously on a mixed group of receptors with an agonist action on one side and with an antagonist action on other side. That's why we call it as mixed agonist and antagonist. So examples are opioids. So we can see in a chart, antagonist affinity will be there but no biological response, agonist affinity will be there and full biological response is shown. Partial agonist, there will be affinity but less pharmacological action. Inverse agonist, there will be affinity but opposite action. So 5 categories of agents we can produce. One is antagonist, agonist, partial agonist and inverse agonist, and another class that is mixed agonist and antagonist. Next type of mechanism actions the drug may show are drugs acting on enzymes. They will act on enzymes. So almost all biological reactions carried out in our body are catalyzed by enzymes. Hence enzymes are very important target for drug actions. Drugs are present which may either increase or decrease the rate of enzymatically mediated reactions. In normal physiological systems, enzymes activities are often optimally set. So the stimulation of enzyme by drugs that are truly foreign substances is unusual. So there are very few drugs which can stimulate the enzyme actions, but maximum drugs will stops or inhibits the action of enzymes that are called enzyme inhibition. So you can see this is an enzyme and this one is a substrate. Substrate will bind to the receptor and the receptor will break or hydrolyzes the substrate and produces the products. This is the normal activity of an enzyme to catalyze the reaction. So by this we can either stops the enzyme or we can stimulate the enzyme. So since, as I told you earlier, stimulation is not possible, so maximum drugs are, it will inhibit the enzymes. There are two types of inhibitions, non-specific inhibition and specific inhibitions. So again, this specific inhibition is two categories, competitive inhibition and non-competitive inhibition. So we will see an animation of enzyme.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.
So first, non-specific inhibitions. Many chemicals and drugs are capable of altering the tertiary structure of an enzyme with which they come in contact and thus inhibits it. Means there will be no specific enzyme. It will inactivate or inhibit all the enzymes present on our body. Examples are heavy metal salts, strong acids, alkalis, alcohol, phenols, etc. they will inhibit the enzyme non-specifically. So you can see here the reactions and inhibition. Next, specific inhibitions means in these drugs inhibit a particular enzyme without affecting others. So the target will be specific enzymes. In this category we have two types. Either we can inhibit the enzyme by competitive or non-competitive mechanisms. As we have seen in the animation, competitive, the drug and the substrate will bind to a particular site on the receptor enzyme and it has competition with them. That is called competitive inhibition. Just we have seen in the animation, that is called physostigmine and neostigmine compete with acetylcholine for cholinesterases. So you can see here in the image, this is your inhibitor and this is substrate which has both same structure, so they will compete. So once the inhibitors bind to the enzyme site, it will stop the reaction. Once it is removed, the drug may bind and again the enzyme will become active. So hence there will be a competition, hence we call it as a competitive inhibition. In non-competitive mechanism, the drug will be bind to another site other than substrate site. So because of this binding of this drug or inhibitor to this site, there will be conformation changes in the active site in which the substrate will bind, so which inhibits the binding of the receptor or enzyme by the substrate. So just we have seen in the animations, the inhibitor reacts with an adjacent site and not with the catalytic site, but alters the enzyme in such a way that it loses its catalytic property. Examples are acetazolamide carbonic anhydrase inhibitor, aspirin indomethacin cyclooxygenase inhibitors. Next is non-receptor mechanisms. We have seen specific interactions like receptors. Next is partial specific like enzymes, and third one non-receptors and non-specific mechanisms. So in this, some drugs do not act by combining with receptors or enzymes. They act purely in mechanical, chemical or other ways. For examples, gastric antacids acts by neutralizing the hydrochloric acid secreted by parietal cells in the stomach. So their action is chemical reaction, that is called neutralizing reactions. Next example is osmotic diuretics like mannitol remove excessive fluid from the body by increasing the osmolarity of plasma, and the glomerular filtration results in diuresis. So this is because of increase in osmolarity. Third one, certain antimetabolites like structural analogues of purines and pyrimetabolines are incorporated into the nucleic acids, disturbing their functions. So they act as antimetabolites. Next, chelating agents like EDT are employed in the treatment of poisoning by heavy metals. So these drugs will act by chelating that heavy metals. So their mechanism action is not an enzyme based or not on receptor based, just they will bind to that drug. Next, drugs acting through antibodies. So vaccines produce their effect by stimulating the defense mechanism in the body and introduce or induce production of antibodies. These antibodies will kill the antigen or virus which is coming to in our body and protects our body. So the drugs basic mechanism is to produce the antibodies in our body. Examples are vaccine against poliomyelitis, that is polio vaccines, rabies vaccine, smallpox vaccine, typhoid and cholera vaccines, while antisera contains antibodies which neutralizes toxins in our body like antisera against diphtheria, tetanus, gas gangrene, etc. What will these antibodies will enter our body and blocks the toxins. So their mechanism is by either by production of antibodies or by binding to the receptor with the help of antibodies. Receptor means toxins with the bodies. And the last is placebo effect. Placebo means I shall please. What happens in a psychological patients? What we will do, we will give an inactive substance to satisfy the patient demand for medicines. So many diseases which are chronic in nature have a substantial subjective psychogenic elements in addition to the somatic elements. So such studies have shown inert medicines like placebo can lead to improvement in more than 1/3 of all persons. This is termed as placebo response. Some doctors use the placebo response to reinforce the effects of treatment. So we will give inactive drug to the patients to satisfy its demand. So what we have learned? We have learned the various mechanism of drug actions. First is receptor based mechanism action, that is called specific mechanism action. Second one is enzyme based mechanism action, that is called as partial specific. Third one is non-receptors or non-specific mechanisms actions. Fourth one is action by producing antibodies, and the last is placebo effect. Thank you.